Hey kiddos, so this is your lesson for Thursday the November 12th and a little bit into Thursday or Friday the 13th. Right now let's recap what we've learned so far. So why do we have to get into this whole cellular division anyways? Why do we even have to worry about it? Well, because we need to have a lot of cells in our bodies. We know that cells are the basic building block of all life, and we are multicellular, so there's a lot of us. Why do we need so many cells? Well, that's because cells can't really get super big, because as a cell gets bigger and bigger, the surface area, that area of the cell membrane, does not increase as fast as the volume, otherwise known as what's going on inside of that cell. So our surface area, that's our cell membrane. That's all the space that we have to get things to go into and out of the cell. The volume, that's everything going inside of that cell. As the cell gets bigger and bigger, the cell membrane can't keep up with the demands of the cell. So those cells are tiny. So that's why we need to multiply and we need to have the ability to our cells to divide. So that way we can have several small cells making up a tissue and then eventually making up an organism rather than one or two big guys, okay? So that's the whole point of cell division. We also need cells to be able to divide because in each and every one of those tiny little cells that we have, there needs to be this guy in there. There needs to be DNA. So we need to make sure that we're making appropriate copies of the DNA so each one of our new cells has that copy. Why also do we need cell division? Well, because we need cells to divide and reproduce so that way when we do cut our cells, we have the ability to heal, to make new cells. That's kind of a big deal. So another thing that's going to be important to remember is the difference between chromosomes and chromatin. This should have been in your reading from Monday, and it definitely was in the gizmo yesterday, but let's review. Our chromosomes, they're like X's. We're going to look at them in a little bit. That's when the DNA is really, really organized. Our chromatin, you're going to think of that as spaghetti when the DNA uncurls and it relaxes into the nucleus. So you should have known from yesterday. Let's look at the different processes that go on in the cell. So interphase is where we find our cell. Most of the time, this is their growing and preparing for the division. This is where they spend most of their lives. You need to know about all three of these stages. The G1 or the gap 1. So this whole process represents interphase. This is what your cell is in. You see this tiny little yellow spot here for M, that's mitosis. That's when we divide. Then boom, we're right back into interphase. So your cell is spending most of their time in interphase. But in interphase, there are 3 separate stages of interphase. G1 or the gap 1 phase, that's just the cells going about their business. They're just being a cell. During the S phase or the synthesis phase, that's when the cell copies its DNA to prepare for cell division. And then in the G2 phase, which you notice is right here, right before mitosis, that's when the cell is preparing to divide. So from all of this, I want you to know that the S phase in interphase, that's when we start to copy our DNA, okay? Now, mitosis itself has 4 different stages. You should know this from last year, but also from your gizmo from yesterday. So how I like to remember the cell cycle is IPMAT, I-P-M-A-T, because I do the I first stands for interphase, and the rest of them, P-M-A-T, PMAT or IPMAT, if you add the interphase, that's going to walk you through the rest of the stages. So looking at your gizmo from yesterday, oops, man, my computer is all over the place today. You have interphase. So see how in here we have this chromatin, right? So it's all that's our DNA. That's what we need for our cell. So it's all wound up together and it's kind of a mess, but it's here in our nucleus. And remember, we have our G1, our S, and our G2 phase. Moving on to prophase, that's when our chromatin condenses and becomes organized and it comes into these little guys called chromosomes. It's really cool because you can actually look at a cell undergoing this, and it forms these little X's. It's so interesting. It's so cool. Um, so this is the first stage of mitosis. Our nuclear envelope, see that dotted line? That's starting to disappear because we don't want our nucleus to be here when cell division is actually happening, because we need to actually split our nucleus into two. Now, you'll notice it says each chromosome consists of two chromatids connected by a centromere. So this is a chromatid, this is a chromatid, and where it stays together, that's a centromere. That's going to come into play in meiosis. So right now, just make sure you know those words, but we're going to dive a little more into that when we go into meiosis. Now, what's going on outside is outside of our nucleus, our spindle fibers are starting to form between our centrioles, and our centrioles have moved to the opposite side of the cell because they are getting ready to help us divide. If you remember, centrioles are organelles that help cells divide. Moving on next to metaphase. Metaphase is really easy because you're going to think meta, middle, because this is the second phase of mitosis. Let's see if I can move my head. The centromeres start to attach to these spindle fibers. So those spindle fibers are attaching to these centromeres right here in the middle. And then the spindle fibers, they start to change in length and they pull the chromosomes into a line. So when you see metaphase, you guys are going to think middle. So on some quizzes, you might have to draw some stages, or you might have to match the actual stage with the picture. So this one's super easy. Just remember that meta, middle. That's when those chromosomes are lined up in the middle. Moving on to anaphase, this is another easy one. Anaphase, apart. That's when those chromatids, they pop apart. So this is our third phase. We have prophase, metaphase, now anaphase. The centromere split. The sister chromatids are pulled to opposite poles. That means to the opposite parts of the cell. And this phase ends when the chromatids, now called daughter chromosomes, reach the poles and stop moving. So we are making daughter cells right now. So this, we have already doubled our DNA. That was what the S phase in interphase was. Now this is our spot in which we pop it apart. So that way our daughter cells are going to have the correct amount of DNA. Anaphase, apart. The last part is telophase. So the fourth and final phase of mitosis. During this phase, the nuclear membrane, now it starts to form. Remember, in prophase, it was starting to break down. Now in telophase, or telophase, either enunciation is correct. Now we're getting ready to make two new cells. So now we want the nucleus to come back. So that's that nuclear and membrane that's starting to pop back up. That's we're getting ready to make two cells out of one. So this is when our chromosomes also start to relax a little bit, and they wind into that chromatin, also known as, or we call it spaghetti, right? Because it looks all stringy and it's no longer very organized. This is telophase is technically your last stage, but you know that you have two cells after you have completed cytokinesis, because that basically means the splitting of cytoplasm. So your cells, and animal cells, and plant cells is a little different. So we'll talk about that coming up here in a couple days. But so what happened? Do you start to see how your cells, we're kind of having two cells here. So the cytoplasm goes to either side to make sure that we have enough cytoplasm in each cell to carry out its processes. But then the cells start to pinch inwards, and eventually we'll have two daughter cells, each containing a complete copy of the parent DNA. So all in all, this is what your cell division looks like. Let me move my head again. You should be able to look at all of these and see how all of these processes go together. There's also a little hand motion that we like to teach you guys to help you remember. And it's pretty funny because I actually, when I teach it, I will see my kids, like as they're taking their tests, they'll use their hand motions to help them. So we're going to go through it together. What you need to know is this is interphase. This is your cell. This is your there you go. This is your nucleus. Life's good. You guys are going through G1. You're doing all your cell processes. And then S phase of interphase. This, why I added my second hand, that's to show that we have doubled our DNA. So make sure you know the S phase of interphase, that's when your DNA starts to get copied. Okay? Then we have G2. We're preparing for cell division. So we move on to prophase because I kind of do this when it comes to prophase because I know that my chromatin is getting organized into chromosomes. So that's kind of how I represent this. So then I have prophase, and then I have metaphase that reminds me that the chromatids or chromosomes are lining up on the middle of the cell. So we have prophase, metaphase, anaphase, remember apart, so anaphase. Telophase, we have two nuclei that are starting to form again, and then eventually cytokinesis. So in here, your hands are representing chromosomes and nuclei. So let's try that again. So here is my cell in interphase. S phase of interphase. I just copied my DNA, so now I have double. G2 phase of interphase, we're getting ready to split. So then we have prophase, everything is getting organized into nice chromosomes. Then we have metaphase, anaphase, telophase, cytokinesis. That's when that cytoplasm splits. So that's just a way to help you guys remember. It's just another way to get this information into your head. Now, for your actual assignment for today, what I need you guys to do is I want to make sure that we have this down and also that you have it in your notes because we actually haven't taken notes over this yet. So if you have your bio book with you, great. If you don't, this is how you get to your book. Go up to your bookmarks up here. Then you're going to go to McGraw-Hill. Then you are going to go down to here. You kind of have to click on McGraw-Hill twice. Make sure you have the right email. Then you are going to go all the way to, I have to click on my bio book. Now, click on your ebook, and you guys are going to go to page 248. I just find it the easiest to do it this way. 248. Oh, come on. It'll get us there. Beautiful. So what I want you guys to do for your notes is one, I want you to read this, obviously, right? And then I want to make sure you know all your vocab words, but also I want you to make a chart in your notebooks in which, kind of what you did on the gizmo, in which you described each stage, but now I want you to make sure you're able to draw and label each stage of mitosis. Now, this one doesn't include, it does include interphase. So you guys are good with that one. Just make sure you put in about the G1, the S, and the G2 phases in interphase. And then once you are finished reading, you are actually, if you're going to stay on pace, you're actually good for today because it probably should take up the rest of class time. But if it doesn't, you can move on to your next assignment, which is going to be this mitosis puzzle, in which you take these puzzle pieces and you're going to fit them all together. Each one of these puzzles has 4 of them, right? So each puzzle is going to have a blue piece, a white piece, a green piece, and a purple piece. It's going to be your job you can move these, so you'll be able to move them around and put them in the correct puzzle. All right, that is going to be, that's going to be due probably by Monday. I might give you the weekend to do it because it might be a little bit tricky. And then I'll post the code on Canvas, but there's also going to be a quizzes that I want you guys to take. Just have some practice problems so we can start practicing questions about mitosis. All right. Other than that, that's all I have for you guys. So I will not be in school Thursday and Friday, but I will be hopefully back ready to go Monday. We'll hit the ground running and we'll move on to our next chapter. So if you guys have any questions, feel free to email me. Other than that, hope everything goes well and I hope to see you all soon. Let's see. And have a good day, guys.